Thank you very much for the invitation and for the kind introduction. I hope I'm audible clearly, and I hope you see my full screen of the front page of the PowerPoint.
Yes, we see.
Okay, very good. So I would like to start a bit with my background. I'm associated with the Institut du Cerveau, ICM, which is, just let me see, yes, it moves. And in English, it's called Paris Brain Institute. So in the team, I am in the movement disorders team, MOVIT, and the Center for Neuroimaging. So it's inside the one of the most largest Western European hospitals, La Pitié-Salpêtrière, and also associated with the Sorbonne University in Paris, where I did my doctoral studies in clinical neuroscience. So I would like to divide also my presentation in several folds. So first fold, I would like to show my expertise, like what diseases I work with. And then I would like to show you the tools we use for these diseases to understand the science behind these diseases and how it could be helpful, particularly on the diagnosis side. We are more involved in the diagnosis side. And the potential for these type of methods, these types of tools, which are becoming very famous for neurological diseases. What is the potential in the neuromuscular diseases? So let's start with Parkinson's disease, a neurodegenerative disorder, quite a multifaceted disorder, manifests as a motor disorder and worsens over time. And the non-motor disorders are also observed, which is also quite seen, like sleep disorders, somebody was talking about as well today. So diagnosis is quite difficult, only possible when the clinical motor signs starts to appear. So from the neuroscience aspect, what I'm interested particularly in is the substantia nigra, which you can see in the animation that it's being highlighted in red. So there is degeneration of substantia nigra, and where there is a dopaminergic neurons, and there is also iron level augmentation in these Parkinson's disease patients. Then another interesting side is the prodromal Parkinsonism, like to those subjects who do not show Parkinson's disease, but they might convert at some stage to Parkinson's disease. So that is related somehow to the non-motor symptoms. That is called isolated REM sleep behavior disorder. So you can see also a sample video from one of our senior heads of department of the sleep center of La Pitié-Salpêtrière from the center of Professor Isabelle Arnaud. So this is IRBD, where it is manifested as a sleep parasomnia featuring unpleasant dreams, such as acting out the dreams in real. And these are characterized by nocturnal violence and increased muscle tone during the rapid eye movement sleep. And it's quite common in synucleinopathy like Parkinson's disease, Lewy body dementia, etc. So these people have a high chance, like majority of them, they convert into Parkinson's disease. And then they start to have these motor signs as well, slowly. So that means there should be mild substantia nigra damage as well, what we see in Parkinson's disease. So at the side of the tool, I would like to show you what is magnetic resonance imaging, which I'm sure most of you are already familiar with. It's from the technology side. It's a non-invasive medical imaging technique that records the changing magnetic field and gives us different kinds of images based on the pulse sequences. That means based on the question, for example, if we are interested to see these neuromelanin in substantia nigra, we'll do a certain kind of image contrast, certain kind of protocol. Then if we are interested in the iron levels in the brain, we'll do a different kind of protocol. And for example, for muscle imaging, for muscle MRI, we'll do a different kind of protocol. So just with this animation, I'm trying to show you how it is. And in this animation, you can see in my right-hand side that how images are captured. So then the mathematical questions can be dealt with, like how many like cubic millimeter, what volume changes, what signal changes, etc. So let's see some examples of these MRI sequences which we deal with. So these are some of the examples of MRI sequences which we deal with depending on the clinical question at hand or research question at hand. So in my case, I'm mostly involved with the neuromelanin type of sequence, which is important for Parkinson's disease and the IRBD, what I showed you, because it's like a proxy marker for dopaminergic neuron changes. Second, I'm more interested is in RN, quantitative MRI. That helps us to get the quantification of iron in the brain and perhaps in other muscle area as well. So this makes us think that there is an urgent need to develop these MRI or imaging biomarkers in the field of disease diagnosis in different kinds of diseases. So this could help in monitoring the disease progression primarily, or analyzing the effects of disease-modifying treatments as well, and can help in early stage of the disease as well. So let us now examine such damages at the level of the structure. So like I mentioned about the neuromelanin pigmentation inside the substantia nigra, which can be quantified using a certain kind of MRI technique. So neuromelanin is a dark, insoluble pigment concentrated in the dopaminergic neurons of this structure, substantia nigra. And interestingly, also in the noradrenergic neurons in locus coeruleus. So from the technology side, it is paramagnetic when it's linked with metals, and when you scan the patients, it becomes hyper intense on these kind of protocols, T1 weighted images. So what does it mean? It means that then it starts to highlight that area where you see the substantia nigra. So you can see in these images, it's a healthy volunteer, a normal subject. It shows all the substantia nigra highlighted, and then you have Parkinson's disease patients starting to lose. So any layperson can see visually that there is a loss of substantia nigra neuromelanin here. So like dopaminergic neurons are starting to lose. So similar for the IRBD as well. So similarly, you could see for the iron changes in the substantia nigra. Like I showed you the sequence QSM. So you pick the QSM's protocol and you are scanning these patients, and then you see that you can quantify iron also in substantia nigra using this QSM technology of MRI. So that could help to compare the patients with the healthy volunteers and see that there is an iron augmentation very easily and non-invasively. Another structure which is implied is very interesting as well, which is locus coeruleus and subcoeruleus. So it's a tiny structure in the bones of the brainstem, and it is also visible using the same technique of neuromelanin. And as you can see here, for this prodromal stage of Parkinsonism, REM sleep behavior disorder, isolated ones, you see that these two tiny spots, one on the left and one on the right, this is locus coeruleus, subcoeruleus, and these tiny ones disappear when they start to have this IRBD. So that is also like a marker for Parkinsonism to understand how these changes happen. So interesting part is to quantify. This is very interesting. When you scan in MRI, after that, you have to use various strong technologies, strong ideas mathematically to segment these regions so that it gives you as precise as possible the results so that we can see the changes. So from the analysis side, if we could see the measurements, how they are done. So when we segment, it gives you something like this. In green, HV is the healthy volunteer. In green, you can see the substantia nigra, same what we discussed. And when the early Parkinson's disease patients are coming to get a scan, you see there is a drop. You can see clearly from my left-hand side, these images, the first row and the second one, you start to see that there is a slight drop in neuromelanin dopamine neurons. And then you see when you start to progress, like this is around 9-11 years of Parkinson's disease. And the second row is around 1 to 2 years Parkinson's disease. So you see that substantia nigra neuromelanin, it starts to disappear. In the lower row, you see without the region of interest. And then afterwards, I'm showing you a TIV segmentation, that is total intracranial volume. You can see the example, how it is done in this MRI. So it helps us to sort of normalize the results so that we don't care about the proportion of the patient. If the patient has a certain head size, or for example, the effect of age, effect of gender, etc. So we divide these values getting from volumes by this total intracranial volume to normalize. So showing an example of locus coeruleus segmentation here as well. So in this is a template where we segmented primarily like an average, and after that you see this is the tiny flash, like left hand side flash of locus, and then you have the right hand side flash of locus coeruleus. So primarily it goes on to find a window. You see in yellow in the right, its right hand side, and red in its left hand side. left side of the subject, and eventually in the window is trying to find the most highlighted pixel. So in the D image, you see red is the left locus coeruleus and yellow is the right locus coeruleus. So this is getting as precise as possible, but it's very difficult, needless to say that. In the second part, you have the iron segmentation here. You see how the nigral iron is segmented in these QSM images. Now let's see about the tools used to segment this, because this is like a very difficult question to find the precision in segmentation. It could be any kind of patient, it could be healthy volunteer, it doesn't matter, but the precision is very important. So that brings us to artificial intelligence. So in artificial intelligence, we particularly use the idea of deep learning. So what is the motivation behind deep learning? It's rather inspired from the human visual cortex, and the biological neuron is the real factor which inspires us to do this. So it's like you need to have little guidance from the programmer. Nobody has to do like proper programming for this, but once the AI is ready, you just send the image and it should just segment right away. That is the basic idea. So one time you program, one time you make it ready, and once it's ready, it has to work on its own and it starts to get better and better. And you can see in this example that how humans perceive a cat in a similar way with like a biological neural network. We try to make the model learn in a similar way with the images, and the model learns. And for example, with the Nigra, it could segment in similar precision as human raters. So what is convolutional neural networks? A convolutional neural network, also called CNN or ConvNet, they are used a lot in the imaging domain for segmentations. So the idea is it's like a feed-forward artificial neural network, and the connectivity patterns, as I mentioned, between the neurons is inspired by the organization of animal visual cortex. So why it's called convolutional neural network? Because it involves various layers inside it, like convolution layer and several other layers. So I would just like to explain you what is convolution layer, why it's called so. So with this example, in this animation, you could see like the brain would learn, and eventually you just send the image in this question, and the model does not know what it is. And just right after learning it using several layers, it could say, okay, whether it's enlarged heart or it's a tumor, it's tuberculosis. And it could right away recognize using this model that, okay, the patient has tuberculosis. That means the model has learned using several images, several MRIs, X-rays, etc., that how to identify these diseases. So let's see how we define these layers. Like, let's see one example of what is convolution. So convolution is the idea is you can see in these layers that is just the idea is to learn by layer using the mathematical convolution operation. So if you have one convolution operation involved, it's called convolutional neural network. So here in this demo, you can see how it scans the image using the filter to conduct the convolution operation. And for example, for tumor segmentation, also it's very similar. You're inputting a tumor, for example, in the MRI, and it goes to several layers, like I mentioned. So first it's called input layer, and you can have one or more hidden layers. It goes to all the hidden layers, and finally in the output layers, it finds you the tumor, what the user is looking for. So it passes through high dimensional MRI input data, multiple layers. Okay, so there is another technique very important in this AI area that is called data augmentation. Why is it being done in the medical domain? Because you cannot have many images for the model to learn. It's not very easy to make it learn with few images. For example, if you are feeding just 50 tumor MRIs, the AI model would have difficulty to really recognize precisely. So then what we do, we get inspiration from other areas of computer vision. That is called data augmentation. So what you see in my right-hand side is these picture of humans. Actually, they are not humans. They are fake photos created from real photos and merged, changed in certain way using all these filters that they start to look like humans. So they are like fake human pictures. So we take inspiration from that. And below that, you see one of my MRI images of neuromelanin, and you see that image is quite distorted. So you take a normal image, you make it distorted. It's like a fake data augmentation technique. So it's an analysis technique used to add the amount of data in an artificial way so that the model learns very, very fast. And it's quite widely used in facial data generation, in face detection algorithms, and becoming very useful, particularly in medical world, when we have very less images to train on. So one of the examples I would talk about, which we did for Parkinson's and REM sleep behavior disorder patients, what we published in Movement Disorder recently, we were trying to study this region, what I showed you at baseline, and the drop in 2 years here. So in drop, you can see clearly and follow up that the nigral neuromelanin starts to disappear. And further, we use this segmentation idea to study the spatiotemporal changes as well, where we characterize the topography over time. As you can see here, there was a healthy volunteer, then studying IRBD and early PD, and eventually for the advanced version of progressing PD, you can see that there is a loss of substantial nigra. So eventual question here is the segmentation, that how to segment as precisely as possible to understand not just the whole structure changes, but also territory-wise changes. And that idea is not just applicable in the brain, but also elsewhere. So furthermore, we took the same idea by manually segmenting these images and having a database of manual segmentation and use it in AI model to create, like by default, precise predictions for new subjects. So for example, what I did here, I used 60 images trained and tried to predict it on 176 by preparing an artificial intelligence model. So it's similar idea, you segment by yourself manually, and then eventually it gives you a result, an AI in fraction of seconds. So here you could see an example of how segmentation is done. So here you saw it's the precision that in left of the subject is in red, and in the turquoise, it shows you the substantial nigra segmentation precisely done by the AI model. So this method we recently published in a methodological journal, Neuromag Clinic, and the neurology side for IRBD and Parkinson's patients in movement disorder. So, so far we saw that it's quite clear that AI using MRI biomarker could be powerful for the brain study, for the neurological study. What are the implications? What are its potential in neuromuscular diseases? Although there is not much work done in that aspect. So muscle MRI can also provide information that is complementary to clinical, histological, genetic, and all laboratory findings we saw for the diagnosis of neuromuscular diseases.
Mute, mute.
Click and say.
I hope you hear me. So muscle MRI allows the identification of various, for example, fatty replacement of muscle tissue, and it's quite common in the diagnosis of, for example, inherited myopathies that are distinctive patterns of muscle involvements on MRI. So these patterns are also established by identification of preferential spared muscles, which may not always be clinically observable easily. So in this way, the radiological implications could be very powerful for these patients. So certain patterns, for example, are specific for a particular myopathy, and others are common to multiple disorders. So let's see some examples, some demos for the implications in neuromuscular diseases. So this is a study recently published in one MRI methodological journal, where they're trying to use AI to segment the thigh and leg muscles, done by a collaboration between Italian and Swiss team. So they are using a protocol very similar to what I showed you in QSM, that is quantitative MRI protocol. It's called GRE sequence. So what they did, they took 6 healthy volunteers and 30, altogether 48 patients, with neuromuscular diseases, and the patients were scanned longitudinally 3 times. The scan time is quite tricky. It is quite long, but in brain MRIs, it's quite lesser comparatively, like it's 5-6 minutes for neuromelanin, which I showed you. But here it took them 20 minutes for the thighs and 15 minutes for the leg. So altogether, they had a huge image data set, as you can see, like the slices of images done manually by the experts. So the idea would be to use all these 770 thighs, 330 leg slices as input for the AI. So let's see an example here. In the upper side of the A image, you see the thigh, and B, you see the legs, and you have all other labels. So this is a proper illustrative example of the MRI scans done there, and their corresponding muscle manual segmentations. So using this, they would start to predict. Let's see how is their prediction result. So you see in my left-hand side, the thigh, and my right-hand side, the leg. So in the training data set, what they use, it was very highly accurate for them. So you see, this is human side in my left, and DNN is deep neural network, DNN. So you can see in the segmentation that DNN could replicate very similar. So DICE is a measure for similarity index, like done by the humans and done by the robots. So what is the similarity? So DICE, you could see for training, is 96.31%, which is quite remarkable, I would say. You see similar for leg, it's above 90. So for case 1, you see, visually, if we see, it's quite similar. Similar you see for case 2, but these are for training data set, the data set they used to train the AI model. Then you see for validation, the ones which were not touched during the software preparation, like during the AI training. So these are images coming from outside, and they are just feeding it in the AI to have the results. So here also you see it slightly goes down, but still it stays more than 90%. And you see case 1 and case 2 and the case 3 as well, not quite bad, all quite high, more than 90%. So I would say quite exceptional result with the overlap between manual and the artificial intelligence segmentations, showing the power of AI in MRI for the neuromuscular patients as well. There is another study done in Australia. It is by using the T1 weighted MRI, is like one of the classical conventional MRI system, like using cerebral palsy patients, involving 20 children, where 6 of them had cerebral palsy. And as it is quite known that the muscles of children with cerebral palsy are smaller than muscles of typically developing children. So that could be a good marker for MRI to compare how it works out. So even this AI model was quite powerful, and they could replicate, as you see, DICE as a similarity index between manual and automatic segmentation of 90. For the typically developing children MRI, and for cerebral palsy of 0.85. So it's still excellent, I would say. So in this example, you could see A is a typically developing child, and B is the cerebral palsy. And there is C and D are the corresponding manual segmentation, respectively, in axial slice, approximately midway between the ankle and the knee. And you can see the precision is quite strong. Like if you see from an expert eye, for as radiologists, is quite remarkable how the segmentations are done. So as it is well known that they differ in shape, position, size, volumes, this could be a very, very strong marker for the future of neuromuscular disease patients as well. So these examples are also very interesting. These are what is produced by the AI models, example segmentations of muscles and bones from MRI scans of the 4 participants, as you can see, 1, 2, 3, and 4 in rows. And these are produced by 6 AI models, which these people used, and along with the human raters. So first, this is the input MRI, the grayscale one, and this second column is the manual segmentation done by the expert, apparently by radiologists, and the others are replicated by AI. So with human eye, we could feel that the replications are quite strong by the AI. And however, this example shows how first and third rows are the scans were kind of not so well segmented, and the second and fourth row are examples of quite a well segmented one. And then I have one 3D rendering images for these. Also, the 3D unit is AI example. Second is also done from AI. Third is also done from AI. And fourth is by manual segmentation. So with these 3D renderings of segmentations of MRI scans, with the AI predictions compared to human raters, you could say, it produces very accurate segmentations, actually. And all these 3D renderings were taken from same viewpoints, indeed. So you saw the potential of artificial intelligence, how it in the domain of neuroimaging, as we do, it can also be used in muscle imaging in the similar way. And it's an area not so tapped into these days by the medical researchers. Final words. So we saw multimodal different types of MRI biomarkers, and particularly coupled with AI segmentations. So we saw it's promising and potentially transferable to different skeletal muscle groups as well. It is direct, and being artificial intelligence is non-rater dependent. Once you have your AI ready, there is no rater implications in that. It's non-invasive because it's MRI. The patients are not quote unquote touched in these cases. So these automated methods can also help monitor muscle growth and evaluate the effectiveness of interventions, and possibly the first step towards pre-symptomatic diagnosis of these diseases one day in clinical practice. Thank you very much for your kind invitation. I really, really appreciate it. And thank you, EAMDA, again for everything. And just a note of thanks for my friends and colleagues. And I'm very much reachable if you would like to collaborate, if you would like to discuss, and if you have questions, even afterwards. I have my website, my Twitter, and my email also here. Thank you very much.
Thank you very much for your presentation. And we open the floor for questions.
Can I make a couple of
Yes,
comments? Not question. Rahul, thank you very much. This is very invaluable. Actually, your presentation is invaluable. I'm a mechanical engineer and a patient advocate since my daughter has a genetic related disorder. So over the past 2 years, I have been, trying to understand what's going on this field. And as an engineer, I have understood that AI is one of the biggest things that have been helping related to gene therapies and all these other stuff related to the genetics. And also I have come across a British-based company, DeepMind. One of the executives is from my university in Turkey, and I have managed to reach him. So actually, this is super important during our journey to be able to reach the drugs about all these genetic related diseases. AI is super important. So thanks again, and I'm glad to see such a presentation and an expert from this field, because I think all patient advocates also have to find and seek solutions to support experts like you on this field, because I'm pretty sure that this will have a big impact on finding the cures, to focusing on AI in the future. Thanks again.
Thank
Also
you
for
very
the
much.
organizers to find.
Thank you. I appreciate it. Thank you.